Hello, so good evening to everyone. I'd like to welcome everyone to Aptamer Group's intro presentation, introducing the company, our technology and our first year results. So we'll start off with a quick introduction to the presenting team, then on to the presentation where we'll do a brief overview of the company and our results for the year ended June 2022. I'm going to do a brief overview or snapshot of the business, and then we'll switch between myself, David and Jenny for scientific and financial inputs. Next slide, please. And next slide. So just a quick introduction to me. So I'm co-founder and CEO of Aptamer Group. I have a background in structural biology and biophysics, and I've run the day-to-day activities of Aptamer Group for the past 10 years. I continue to play an active role in the scientific and commercial aspects of the company, sitting on the scientific advisory board alongside David, both having world-leading scientific background in aptamers and related technologies. David.
Thanks, Aaron. Hi, everyone. My name is David Bunker. I'm the Chief Technical Officer and one of the other co-founders of Aptamer Group. I'm a molecular biologist by training and have now over 20 years experience developing aptamers against a wide variety of targets for almost any sort of application you can think of. Jenny?
Thanks, David. Hello, I'm Jenny Cutler. I joined Aptamer Group in June as interim CFO. I'm a chartered accountant and have previous experience in other AIM and main market companies.
Right, so in terms of a business snapshot, as a company, what we do is we solve what we term as intractable problems for pharma and biotechs by disrupting the affinity ligand market, which Dave will come on to explain in the next few slides, with the development of molecular binders called Aptamers. And we do this through our highly automated discovery platform. Automating the science in the way that we have leads to higher project success rates and faster completion of projects. And this has led to us attracting 75% of global top 20 pharmaceutical companies. We listed the company on AIM in December 21, raising 10.8 million. And we've increased revenues 150% since doing so to 4 million, in line with expectations, which was a key and important milestone in our maiden year. As of June 22, we retained 6.7 million of cash. We've relocated to new premises from circa 7,000 square feet to now 18,000 square feet. We've increased our headcount by 45%, and we currently stand at around 57 people. We continue also to keep Aptamer at the cutting edge of science with the development of next generation Aptamer binders, which Dave will explain a bit more about later in the presentation. So at this point, I'll hand over to Dave for an introduction and background to the technology.
Thanks, Aaron. So as Aaron said, Aptamer Group operate in what's referred to as the affinity ligand market. So that's a market that's worth about $146 billion currently and growing at a healthy rate of about 11.3%. Now, firstly, we need to say, well, what is an affinity ligand? So everyone will be aware of COVID and the lateral flow test that we all got used to doing. Those lateral flow tests rely on what is called an affinity ligand. It is essentially a binding molecule that, in that case, captures the COVID virus and allows it to be detected. So tests of that nature and many therapeutics rely on the ability to generate these affinity ligands, these binder molecules. Historically, the affinity ligand market has been dominated by technologies like antibodies that you can see on the left-hand side of the slide there, represented by companies such as Aptamer. Now, that technology dominated for many years because there weren't many alternatives, but there are well-known issues with antibody-based technologies, and I'll come back to those in the following slides. But recognizing those shortcomings led to the formation of other companies with other technology platforms, as you can see going across the middle of the slide towards the right-hand side, such as the recombinant antibodies or other protein scaffolds from Argenx, Avacta molecular probes, etc. Aptamers also fit into this space, but aptamers are quite unique because all of those other technologies are protein-based and they have their own limitations associated with that. Aptamers are nucleic acid based, and I'll come back to that in a minute, so they are quite unique within that space. Now, aptamers are an important disruptive platform within the affinity ligand space, and themselves are currently positioned at about 2.5 billion market, growing at 20%. So you'll see that's higher than the affinity ligand market itself, due to the attractive characteristics of aptamers over other technologies. If you skip on, please. So briefly to describe what an aptamer is. So an aptamer is a short synthetic, that's an important point, piece of DNA or RNA that binds to a target of interest. So if you think of COVID, we could be binding to the COVID virus itself, or anything that a customer may need a binder molecule to. Now, if you think back to high school or the sci-fi movies, you probably think of DNA as this double-stranded molecule that you can see on the left-hand side. Now, DNA doesn't just exist as that double-stranded molecule, it can also appear as a single-stranded molecule. So if you think of this as two strips of Velcro stuck together, you can peel away one side of that molecule to leave a single strand with a load of hooks that are exposed. In the middle of the slide, you can see how that Velcro strip may fold up into all sorts of complicated shapes, where some of those hooks are involved in forming structures, other hooks are left exposed, which can then interact with all sorts of other targets. So this is how an aptamer actually works. And you can see on the right-hand side of the slide there, an aptamer or an optamer in blue interacting with a customer's target shown in purple. So there, the aptamer is performing as an affinity ligand. Next slide, please. So as Aaron said, Aptamer Group see ourselves as an enabler of technologies. We develop these binders for other customers to help them overcome their respective problems in their fields. We do this because it's well known that approximately 50% of research antibodies fail to meet their performance criteria. So they either don't bind at all, they don't bind well enough, or they cross-react with other things. So it's sometimes not possible to get an antibody-based affinity ligand. There's also the ethical concerns around the antibody space, in that the vast majority of them are generated in animals. That also makes them very expensive to manufacture and difficult to scale up in terms of their production. They're also not particularly stable molecules, so they can be difficult to store and transport. Our Aptamer platform, as Aaron mentioned, is a high-throughput, highly automated development platform that allows us to handle multiple targets in parallel and allows us, therefore, to reduce the human error in the process because robots are handling it. We can handle many, many targets at once because all of this is done on liquid handling robotics. And it also reduces the delivery time. And importantly, there are no animals involved at any stage in our development process. You skip slide, please. So to describe a little bit about the Aptamer platform and what makes us different, you can see on the left-hand side of the cartoon there, an Aptamer. This is the full-length parent molecule that we start with in our process. We develop an Aptamer, which is the minimal functional fragment that retains the ability to bind to the customer's target of interest. Doing this minimization process gives us a number of advantages in that the molecule itself is smaller, which often helps it perform better in many applications. It also makes them more cost-effective to manufacture and more readily scaled. Now, we're going beyond that to develop what we're calling Aptamer Plus, where we are incorporating modified nucleotides into our Aptamer binders to increase the repertoire of molecules that they can bind to and to give them additional functionalities. And we'll come back to that later on. If you skip to the next slide, please. So as mentioned, the process is highly automated, which gives us a number of distinct advantages, mainly around the fact that the process has got a higher throughput, it's got a higher success rate because there's less human error, there's less failure. This platform is also more difficult for others to replicate because there's a lot of time and know-how has gone into the development of that platform. So if another company wanted to do this, they would have to invest very, very heavily in a similar platform. So they would be years behind us anyway. And it also gives us an advantage that when we need to scale, we can replicate that platform and get another team to run alongside it so we can scale much more readily than other platforms. If you can skip to the next slide, please. So where are we actually operating? As you can see down the left-hand side, we mentioned some of these companies before. They all operate within the affinity ligand space. Now, the affinity ligand market can be broadly broken into four key verticals or buckets that you can see across the middle of the slides there. So that's the research reagent market, the bioprocessing and purification area, the diagnostics that we mentioned in terms of COVID, and therapeutics, for example, drug delivery vehicles or novel chemotherapeutics. So each of those affinity ligands down the left-hand side generally operate in one or two of those verticals. But as you can see across the bottom of the slide, Aptamer Group are operating and are making revenues in each of those four verticals. So we have projects in each of those project areas. And that's a good segue into the next slide, and I'll hand back to Aaron to tell you a little more about how we generate revenues from each of those verticals.
Thank you, Dave. It was a great introduction to the technology. So as Dave said, the affinity ligand market is valued at around 145 billion and is growing at around 11%. As a business, we operate across three broad areas, selling services into the four broad buckets that Dave mentioned. Inventively, the names of these companies are Aptamer Solutions, Aptamer Diagnostics, and Aptamer Therapeutics. And it doesn't take much of a stretch to understand where these businesses are positioned. Aptamer Solutions focuses on the custom development of aptamers. Aptamer Diagnostics focuses on the application of aptamers into diagnostic platforms. And Aptamer Therapeutics is focused on the enablement of tools for the delivery of drugs in a therapeutic context. This is a reduced risk model, particularly where the contract research services are contained within Aptamer Solutions. We get paid for what we do in terms of our problem solving, and there is no exposure to customers' R&D. And because we retain the intellectual property, there is potential upside when the technology is commercially exploited by our customers. In addition to this, because we enter the development process, often when there are problems, we enter the process at a later stage than if we were to develop diagnostics and or therapeutics ourselves. So we are closer to a stage at which royalties and milestones will typically be paid. So the key take home from this slide is that we operate a unique platform technology driving three business models, generating revenues in the near term with potential for substantial value creation in the longer and medium terms. Next slide, please. So we'll now talk about some of the operational review and the progress since IPO. Next slide. So we introduced the technology, explained the revenue model. So I'd like to talk a bit about our growth strategy. As stated, we recently listed on AIM and raised 10.8 million of funds. This has raised our profile as a business and more importantly, given us growth capital. So our first area of exploitation of the funds is in the scale up and automation of our platform. And this enables us to improve automation capability and remove operational bottlenecks. The second area is the investment in our novel nucleotide chemistry platform, which Dave mentioned, Aptamer Plus, to develop a differentiated and proprietary offering for us as a company. The third area is the expansion of our commercial team and the development of expanded service offerings as a business. And the fourth area is the use of some funds as working capital while we deliver our strategic plans. So next slide, please. So we're particularly proud of the relocation to our new headquarters, now boasting 18,000 square feet of state-of-the-art labs and office spaces to house our expanding team and automated solutions. So here are just a few pictures of the labs and offices. As part of the funding is to rapidly grow our team, it's important that the investment is applied to our infrastructure, and this has enabled us to increase the square footage and also improve the efficiency in which we can deliver services to our customers, and allows us to have that additional capacity for the increased demand for our services. Next slide, please. So we continue to expand on the commercial front. So one of the big focuses for us is strengthening our team, which we recognise is key to our success. And in that connection, we've appointed Jenny Cutler as interim FD and Alistair Fleming as Chief Operations Officer. So since our year end, our head count has increased from 49 to 54 and is now currently at 57 members. We've grown our sales and marketing team from a team of 2 to 10. We passed our ISO 9001 audit and expanded our quality team to ensure we are delivering top services to our customers. We continue to grow our assay development team to effectively build additional services to offer to customers. And we have appointed Dr. Shozo Fujita, an international expert in oligonucleotide chemistry, for the continued development of our novel Aptamer Plus platform. So on that neat segue, I'll pass over to Dave to discuss some scientific update with regards to our new platform, Aptamer Plus.
Next slide, please. Thank you. Yeah, so you'll remember this cartoon from a few slides ago. As Aaron said, we've made some progress in this area. So we have developed some of the chemistries necessary to generate the Aptamer libraries and incorporate these modified nucleotides into our platform. So as Aaron said, this will provide a strong differentiator for the company, because other Aptamer companies or the developers of Aptamer-based technologies will not have access to this platform, so customers will not be able to get this from anywhere else. So this is truly unique to Aptamer Group. If you skip to the next slide, please. So this slide just summarizes where the Aptamer Plus platform will fit and why we've invested in this technology. As you may remember from a few slides ago, some of the advantages of the Aptamer platform over other technologies like antibodies are listed down the left-hand side, in that we can address different sorts of targets than antibodies. We have a more ethically sound, faster, and more scalable development and discovery process. The manufacturing of our Aptamers is entirely synthetic. There are no plants, animals, or bacteria or anything like that involved in their manufacture, and they're much more readily shipped, so they are more stable. So you can see across the top the antibodies on the left-hand side and the associated problems with those, how the Aptamer platform across the middle improves on those criteria, and then our expected performance criteria from the Aptamer Plus, essentially solving any remaining problems within those spaces. So this is truly a game changer in terms of platform for us. If you skip to the next slide, please. I'll hand back to Aaron for some key highlights.
Brilliant, thank you, Dave. So I'd like to touch on some key business highlights since the IPO in December 21. So for brevity, I'll just give one example from each area. So Aptamer Solutions continues to secure contracts, one in particular with a top 5 global pharma company, developing Aptamer solutions as quality control reagents for novel neurodegenerative diseases. In Aptamer Diagnostics, we've continued partnering and developing molecules to go into diagnostic devices, particularly with a company called Proaxis, where the aim is to develop binders to replace antibodies in respiratory diagnostic assays. And with Aptamer Therapeutics, we have extended our collaboration with a Korean company called Pino Bio, identifying Aptamers against several targets to develop 4th generation targeted chemotherapeutic Aptamer drug conjugates. So next slide, please. So since our AGM, we continue to sign deals and move exciting science forward. As I mentioned, we've moved our offices into purpose-built laboratories, and this was completed, allowing us to scale our automation and improve operational efficiencies and deliver better services for our customers. Aptamer Solutions continues to sign deals. Multiple contracts have been signed with top 10 pharmaceutical companies, developing Aptamer solutions for bioprocessing and the support of purification of novel therapeutics. We've signed contracts with another top 10 pharmaceutical company to develop Aptamers as immunohistochemistry reagents, supporting pipeline assets and early stage discovery for pharmaceutical companies. And with Aptamer Diagnostics, we've signed another deal with a company called BioLiquid Innovative Genetics, supporting their development of novel prenatal and placental diagnostic technologies. Next slide, please. So Aptamer Therapeutics, an update since our AGM, we've continued the second phase of our collaboration with cancer research, and this is the development of the delivery vehicle portion of a bispecific therapeutic Aptamer for the treatment of myeloid of CMML and other myeloid malignancies. We've also signed an early stage deal to develop Aptamers to block the activity of naturally occurring antibodies within the body as potential therapies for transplant rejection. Within Aptamer Solutions, we have a significant deal with a biomarker services company developing Aptamers to support multiplex biomarker assays. And we've recently signed a deal with a company called Novavax, which is a vaccine developer, improving the selectivity of their assays. So can you pass on to the next slide? So I'll pass over to Jenny now for some financial highlights.
Thanks, Aaron. Thank you. So this slide shows the financial performance of the group for the 12 months to the 30th of June 2022, compared to the 15-month prior period. Our key performance metrics were in line with market expectation, and we're pleased with these results for our first year as a listed business. Group revenue was 4 million, 2.4 million ahead of the 15-month period to the 30th of June 2021. This growth reflects strong operational delivery of contract research work for customer projects across our 3 business units. Gross profit margin has improved from 42% to 67% year on year as the group utilised available operational capacity to deliver our revenue. This improvement in gross profit margin, offset by an increase in admin expenses of 1.3 million, resulted in an adjusted EBITDA loss of 1.7 million for the year, compared to a loss of 2.4 million last year. The increase in admin expenses is the result of the group's investment in commercial and production teams, and this is reflected in the headcount increase of 44%. If we can turn to the next slide, please. So this shows the financial position of the group. Net assets at the 30th of June 2022 were 8.1 million, of which cash was 6.7 million, and includes the net proceeds from the placing. The increase in the right of use assets and corresponding liability is primarily due to the signing of the new lease at York Science Park pre-year end. We can turn to the next slide, please. This slide shows the group's inflow in cash over the 12-month period. The group had a net cash outflow from operations of 2.4 million, which represented the adjusted EBITDA loss of 1.7 million, an outflow in working capital of 1.3 million, offset by an R&D tax credit of 0.6 million. The group also invested 0.4 million in assets and received 9.6 million from the placing and spent 0.5 million on lease payments. So thank you. I'll hand back to Aaron now for the next slide.
Thank you. So this is just a quick couple of slides for a summary. I think it's fair to say that we have continued strong growth, and we're demonstrating the market need and acceptance of the technology, giving us confidence in revenues being in line for expectations for the next financial year. We continue to build strong traction on a global level, winning contracts with global key players, and we continue to invest in our novel technology platform, developing cutting-edge R&D and intellectual property for the business. And one of the key things that we're looking to do moving forward is development of exemplification data demonstrating the performance of our lead candidates as therapeutic delivery vehicles. Next slide, please. So I think just to summarise, the key takeaways from our presentation are as follows. We've hit our maiden year number of 4 million. Additional capacity and state-of-the-art labs have been developed and built. We've hired extra people and extra operational support. We've expanded the commercial team, strengthened our financial team, and increased our focus on R&D with the development of new products and product lines to support our pipeline moving forward as a business. We have exciting progress in the development of the therapeutic arm of the business with multiple partnerships and building exemplification data packs for use of Aptamers as therapeutic delivery vehicles. So from us, that concludes today's presentation, and we'd like to open up for questions from the floor.
Right, thank you very much, Aaron, David and Jenny. Yeah, we've already got quite a few questions coming in, and certainly There were lots of interesting things you said there. It was quite a trip through an awful lot of stuff. So I suspect we'll be getting quite a few questions here. First one up, I've got at the top of my list at the moment. And do remember, everybody, to give the thumbs up, vote for any question that you particularly like, so it does come to the top of the list. I'm starting with one from Kate here. It says, what was the outcome of your recent roadshow with Liberum, I can never say that, and what else are you doing to attract institutional investors?
Take that. Yeah, I think the general feedback that we've had as a team and as a business has been very positive from our existing institutional investors. They're happy with progress, and effectively the feedback that we have is to keep doing what we're doing. In terms of how we plan to get in front of other institutional investors, well, we're here giving this presentation to give an update to the market, and also we will be planning to do more roadshows and effectively spread the message and the word about Aptamer Group through interactions with other investment houses.
Okay, thank you. Tim says, which of your 3 divisions, solutions, diagnostics or therapeutics, will be your biggest driver of growth for the next 5 years?
Okay, so that depends on the context. So in terms of revenue generation in the short term, that will be Aptamer Solutions, and that acts as a kind of a horizon scanning platform where we can see all of the trends in the market, all of the problems that big pharma are effectively looking for us to solve. The bigger areas for the business are obviously in the therapeutic delivery space, where large upfront payments and downstream royalties and milestones is where the company would like to aim. Now, that I need to be very clear on that we are not looking to develop therapeutics as a business. We're a tools business, and we're looking to enable the development of delivery systems as a service to the pharma industry. And that's where I think the biggest revenues will come from in the fullness of time.
Okay, thank you. Another question from Tim here. Do you anticipate raising further funds in the next 1 to 5 years to fund your growth plans? And if so, do you have any headroom left to issue EIS or VCT qualifying shares as part of an equity-based fundraise? I guess that might be one for Jenny.
Yeah, I can take that. So we only listed in December 2021, so we're still a young business, and I think I'd say we're well capitalized and well funded right now to deliver the strategy that we set out at IPO. In terms of headroom from a VCT point of view, we do have headroom on that. But at the moment, well funded, well capitalized to deliver on those strategic objectives.
Okay, thank you. I'm aware that Jonathan asked, I'm aware that Repligen purchased Avitide a while back. Is Avitide a suitable comparison company for Aptamer's capabilities?
That's a good question. I believe Avitide is the plasmid purification. Okay. It's a very good question. As a suitable comparator, I'm not 100% certain because I don't believe we operate in the exact same market space. I would actually need to refresh my memory on exactly what Avitide do.
You mentioned you've got IP protection for your Aptamer technology. Are there any other lookalike companies in the Aptamer field then at this stage, or do you believe you've got a unique position there because of your IP?
There are several competitor Aptamer companies out there, but what we believe is that we have the most unique Aptamer selection methodology, having automated the process of Aptamer selection, leading to higher success rates than we believe our competitors have.
Mm-hmm. So they could compete with you, but producing the same or lookalike Aptamers, but they wouldn't be able to do it as efficiently and effectively. Is that
that's correct.
Kind of it?
Yeah, so one of the reasons for the investment and the development of the Aptamer Plus platform is in order for us to innovate and give us a truly novel hybrid technology that sits somewhere in between a nucleic acid aptamer or Aptamer and a protein. So the modifications that David mentioned previously in the presentation are actually amino acid side chains that give the Aptamers additional functionality to give this hybrid molecule.
Okay, thank you. Tim's asking, the pharma market is currently dominated by blockbuster multi-billion-dollar antibody therapeutics, for example, PDL-1 antagonists for oncology. Is there any reason why Aptamer-based therapies could not take a large part of that market in the next 10 years, in your opinion?
Well, we definitely hope so. I mean, that's kind of the game that we're in, supporting the development of those combination molecules like alternatives to antibody drug conjugates. Where I think that this technology has a particular unique advantage is in the delivery of oligonucleotide therapeutics such as siRNAs and antisense oligos.
Okay, thank you. Jonathan asks, can you give us a feel for the level of royalties available to you in the diagnostic, therapeutic and purification filtration? In brackets, he says, thinking of your partnership with BioWorks here, segments.
It's a difficult question to answer because the downstream payments are structured into our contracts and many of the customers prefer that we don't disclose terms. In terms of a feel for revenues, for example, if we were to develop a solution for the bioprocessing of AAV vectors, then you could imagine, if you look at that market size, significant revenues from us as a business, for us as a business.
Do you have a particular approach to how you structure a royalty deal? Is it fairly consistent across the piece, say in the diagnostic sector or therapeutic sector?
That depends on the individual customer. Some customers are avoidant of royalties and prefer upfront milestones or upfront success fees. So we tend to tailor the structure of our deals in line with our potential customers and customers.
Okay. What's roughly the split upfront stage payments or whatever versus ongoing royalties? You give us some feel for what your expectation is from each in terms of contribution to revenues?
Well, again, that's bespoke to each individual project. So
I was thinking of across the piece, right? You know, across your present client base rather than particular contracts, as it were.
It's difficult to answer directly, and this isn't an evasive answer. If the companies are smaller and earlier stage, they prefer the deals to be backloaded on success, and if the companies are bigger, they prefer the deals to be upfront and they have more control over the intellectual property. So there is a true variety in the different structures and levels of royalties and milestone payments that come across our desk. Okay,
thank you. Jonathan also says, given that you would typically be engaged in the event that a monoclonal antibody isn't available, are you able to command premium pricing relative to monoclonal antibody providers?
In some cases, yes. Typically, what happened when we first set the company up was that customers would come to us after trying antibodies and various other technology platforms. So we are able to therefore, command a premium if it's a larger company. If it's a smaller company, it comes back around to the structure of the deal, where we may have taken a view, or we may take a view to support the company for a better back end of the deal if it's a particularly exciting technology.
Okay, thank you. Niels would like to know if you have any comments on achieving target specificity for marker proteins, which are themselves capable of binding RNA and single-stranded DNA, such as the SARS-CoV-2 nucleocapsid.
Yeah, I'll take that one. And it's an interesting question. It does come down to the selection strategy, because yes, as you can imagine, nucleic acid binding proteins, while they sound like they would be very easy to generate an aptamer against, generating a specific one can be more troublesome. But that's where the experience of the team comes in. So part of the advantage of our platform is not only can we handle multiple targets at any one time, we can also carry out selections under multiple different conditions at any one time. So we do have the ability to tailor our selection approach based on the requirements of the end target. So we can deal with that sort of issue.
Okay, thank you. In the field of diagnostics, are you being asked to make affinity ligands for established diagnostic biomarkers, or will the scientific need for measuring the biomarker have to be established, which could take many years, perhaps?
I can take that one if you want. Yeah, we've got quite a lot of interest in that area, particularly where the detection is currently done by technologies such as mass spec, which is expensive and time consuming, where people want a quick readout of a molecule that may be difficult to generate an antibody to. Then we do have quite a lot of interest, particularly in the area of small molecule or metabolite detection.
But typically would those take longer to develop as a result?
No, for us, all of the targets that come into the company go through one of three processes. So we have three individual Aptamer selection methods, one tailored towards cell-based and tissue targets, one based to proteins and peptides, and another process for small molecules. So all of the processes have, with the exception of the cell-based selection methodology, the other two processes run through a strict timeline. So all of the targets that come in complete roughly at the same time and over the same time period.
Okay, thank you. Are you concerned about the impact on share price once the founder investors are released from their lock-in period? I should think it was 12 months, was it, the lock-in period?
I think it's fair to say that we have a robust and faithful investor base, and we don't think or expect that the release of the investors from the lock-in will have a dramatic effect, but time will tell. Obviously, we have no control over anything that happens in the market after the lock-in expires. But
sure.
as founders, we certainly have no intention of selling shares.
Right, okay, thank you. Paul asked, when used as a therapeutic, how do you prevent the nucleic acid aptamer being broken down by host enzymes?
Yeah, can do. It's a very good question. It's also a problem that has been addressed many years ago. So other modified nucleotide technologies have been introduced over the last 20 years to address that problem. So those nucleic acid modifications essentially prevent the nucleases that you're referring to from recognizing those targets as a substrate, so they don't get turned over as efficiently. So essentially they're more stable.
Yeah, and if you look at current aptamers that are going into clinical trials, some of those are DNA based with no protection, and I suppose it depends on the application. So if you were looking to deliver a radionuclide therapy or something where a short acting binder is required, as long as the aptamers are stable for a defined period within the body, degradation isn't actually a problem. It can be considered a benefit for clearance.
Okay, yeah, thank you. Would Aptamer therapeutic agents be able to cross the blood-brain barrier to treat CNS disorders?
That's a very good question. I'll hand that one over to Dave. I know this is an area that he enjoys.
Simple answer is yes. It's actually already been demonstrated as a proof of concept. So there are aptamers that target proteins that are known to shuttle across the blood-brain barrier, and the aptamers there essentially piggyback on those proteins, crossing the blood-brain barrier and taking whatever cargo has been attached to them. So yes, it is possible. Yes, it is already been demonstrated.
Right, thank you. Final one here from Robert. Are aptamers used for therapeutic delivery subject to the normal drug approval process?
Yeah, I think all new molecules and new chemical entities have to go through the same process, and then Aptamer or Optima drug conjugate would be no different to that.
Yeah. Okay.
In fact, we actually believe they are the next phase of delivery vehicle for the drug delivery space.
Yes. Yeah. Okay. Yeah. Yeah. I'd have guessed that myself, and I'm no expert. We've got no more questions. If anybody's desperate to ask one last question, we have time, so type away. But if not, we will we'll bring matters to a close. And I'd like to thank Aaron and David and Jenny for their excellent presentation with plenty of information there. For those online, you will be able to pick up a copy of the presentation shortly. It'll be on our website, along with a recording of this webinar as well, if you want to go back around and check on some of the detail. There was quite a lot of good stuff in the slides there, so do check later on our website. In the meantime, thank you to the Aptamer team for joining us today, and look forward to keeping a close eye on development. Sounds like you've got a pretty exciting technology to develop there, so I wish you well with that, and maybe look forward to seeing you again next year, and you can update us on progress. Thanks very much.
Thank you.
Thank you.